Okay, so this is part 3 of my discussion on my first quarter grade 12 biology reviewer. So we've already talked about cellular respiration. Right now, we're just going to discuss related topics that are related to it. So I believe what we're seeing right now are macromolecules. So we have carbohydrates, lipids, and proteins. There's a reason why usually the fourth one is nucleotides. It's not part because, well, there has to be a reason. I can't recall the reason as of now. But yeah, what we have here, carbohydrates, we have monosaccharides, disaccharides, and polysaccharides. One example of a monosaccharide is glucose. From the prefix mono, that means just one. For disaccharide, we have lactose, maltose, and sucrose. For polysaccharides, 3 or more, we have starch, cellulose. Cellulase is what enables the digestion of cellulose. Usually that's found in cattle when they eat grass, because grass basically consists of cellulose. There's glycogen and there's peptidoglycan. We have lipids, there are 3. Fats, phospholipids, and steroids. For fats, it basically consists of 3 fatty acids and 1 glycerol. We have 3, perhaps just 2 common types: saturated and cis unsaturated. Actually, let's just say 3 types: saturated, cis unsaturated, and trans unsaturated. 2 are unhealthy. It's saturated and trans unsaturated. This is why we hear the term called trans fat. So it comes from here. Trans unsaturated fat, that's the complete term, and it's basically hydrogenated oil. I think that's the term, if I recall correctly. So if oil is hydrogenated, that makes it also, let's say, hydrogenated olive oil, if that's even a term, just in theory. If it's hydrogenated, it used to be unsaturated fat. It used to be healthy, one such as olive oil, but because of the hydrogenation process, it becomes trans unsaturated. Cis, this is for example, olive oil, what else? Avocado, foods such as that. Healthy fats, basically. Saturated fat, this is what you see in fast foods. This is the reason why usually if you consume a lot of high fat foods, such as fast food, you have a higher risk of getting something called atherosclerosis. So this is basically the buildup of plaque, buildup of fat in your blood vessels, for instance, the artery. And that can impede blood flow. A lot of problems can happen. It's similar to coronary heart disease. It's just for coronary, the fat is at the heart itself, so that's dangerous. All because of saturated fat. It's not liquid at room temperature, it's solid, and that's what makes it dangerous. Unlike cis unsaturated, this is liquid. So when the fats flow through the blood vessels, there's an unlikely chance of developing atherosclerosis if it's cis unsaturated. Okay, I've discussed a lot on this part. Let's move to phospholipids. Phospholipids, so this consists of 2 fatty acids and glycerol. I don't recall the function of this one. I remember. That is I remember now. In a cell, we have what's called the plasma membrane, right? Let me just find that page again. Something that I've shown to you in the previous video, more specifically part 2. Hang on. I'm looking for it. not here. I wonder why it's not here. Ah, back page. There you go. So essentially, this is the phospholipid bilayer. This is what the plasma membrane looks like in if you magnify it in a way. So yeah, heads, tails. We have the what do you call this? Hydrophobic. I think it's hydrophobic inside, and it's hydrophilic on both ends. Why am I saying that? It is important. I might be able to explain why later. Now, going back, steroids, one example is cholesterol, LDL, stuff like that. And we have proteins, monopeptides, dipeptides, and polypeptides. I still remember two terms, dehydration reaction, and I don't recall the other one. It's just one of them is the process we're in, coming from, for example, a monosaccharide. You add more, and it becomes, well, if you add one, it becomes a disaccharide. I know there are equations for this. Like, for example, glucose plus glucose, that's something. I hope I have it in my reviewer, like the equations, but I can't be sure about that. Okay, steroids, you have cholesterol. Okay, I've discussed that already. Okay, the other term, I'm not sure if that's dehydration reaction. Coming from polysaccharide, for example, you remove, then it becomes a monosaccharide. Yeah, just wanted to let you know about that, although it might be not as useful. Okay, what is this? Cyclic electron flow. I think we're talking about in this page. Ah, okay. You know, let's discuss this later. Yeah, this is about plants. Calvin cycle and light reaction. What is this? Okay, let's talk about this one. Although it's kind of redundant already, I've already discussed this. I guess I wrote a separate page to reiterate certain important points regarding the lesson cellular respiration. So again, you have 4 steps: glycolysis, pyruvate oxidation, Krebs cycle, and I guess the more relevant way of saying or referring to stage 4 is oxidative phosphorylation. That basically consists of ETC and chemiosmosis. So glycolysis happens in the cytosol. The rest happens within the mitochondrial matrix. For more specifically for stage 4, it happens in the inner membrane. Okay. Pyruvate oxidation, this is where glucose is converted into pyruvate. So this is an oxidation reaction. In the Krebs cycle, acetyl-CoA is oxidized somewhere in the early stages, perhaps maybe between 3 or 4, I think. I can't be sure. Do you want to look at the Krebs cycle again? Yeah, I have it right now. Let's see where acetyl-CoA is oxidized. This is succinyl-CoA versus acetyl-CoA. Not between stages 3 and 4, steps 3 and 4. Here, in between 1 and 2, or more specifically step 1. So acetyl-CoA becomes CoA. This is the oxidation that's being talked about here. So acetyl-CoA is oxidized to 2 molecules of carbon dioxide. This is evident in what stages? Here, stages 3 and 4. Always remember two CO2s. That's the end product of the oxidation of acetyl-CoA. And electrons are released from the reaction. Um, it reduces NAD plus and FAD into NADH and FADH2 respectively. So there's a reduction reaction to convert the ones with plus into the one with H's at the end. For step 4 or stage 4, there's an oxidation of FADH2 and NADH. Why is this done? Why are these guys reduced? This is to give the electrons to the electron transport chain in order to trigger chemiosmosis. So this comes along the accumulation of H ions in the extracellular matrix. Either that or the cytosol. Basically, an electrochemical gradient would be formed in order to allow, to let the ATP synthase open its doors for them. And once they enter, eventually chemiosmosis would be triggered. That's the process behind that. Okay. For the reaction, 1 glucose plus 6 oxygen atoms. Atoms or molecules? I think atoms. Yeah, atoms. Plus 6 carbon dioxide molecules plus 6 water molecules plus energy. This energy is through heat or ATP. ATP, or in other words, adenosine triphosphate with 3 phosphate groups. This provides energy to drive endergonic reactions and thus by the transfer of a phosphate group. So ADP plus P is equal to ATP. This is what happens in the energy payoff phase. In the energy investment phase, it's the other way around. ATP minus P, that's equal to ADP. Why is it called an endergonic process? This one. Well, it's because it takes energy to work. So yeah, I won't explain deeper than that. ATP plus ATP is ADP plus P plus energy. This is exergonic if you orient it the other way around, or in other words, releasing energy. Okay, how about the two types of phosphorylation events? This was already mentioned earlier, but not in a way of saying that they are types. No, I mean, not in a way that I said they were events. So two types of phosphorylation. We have substrate level and oxidative. For substrate level, this happens in glycolysis. I think also in citric acid cycle. Not just in glycolysis, but yeah, I think it's more popularly known to be associated with glycolysis. So 2 ADP gets converted into 2 ATP. In oxidative phosphorylation, this happens in chemiosmosis. So in the electron transport chain, ADP plus P is equal to ATP. Again, these are two important things to know. Oxidation, increase in oxygen, decrease in hydrogen ions, decrease in electrons. Decrease in oxygen, that's a decrease in oxidation and consequently an increase in reduction. An increase in hydrogen atoms, not sure, maybe it's ions, as well as electrons, increase in electrons. Okay, so this is endergonic, this one. But one example of exergonic is cellular respiration itself. It's an exergonic process since it releases energy. Okay. In the electron transport chain, we have electron transport and chemiosmosis. Here, we have oxidation. So this is the oxidation of electrons uh, to carry NADH and FADH2. Sounds fishy. But that's definitely correct. I'm just not sure how to explain it properly. In chemiosmosis, we have phosphorylation. This is where ATP is created. So through the electron transport's role of, well, these electron carriers, um, they transfer the electrons so that ATP generation would be triggered. Again, O2, there's a specific description for this. I can't recall. But you have to know that it's the most electronegative in the context of this lesson. And it pulls electrons from the ETC to itself. So as electrons flow from carbon to oxygen, energy is released. So this is glucose. Electron journey. Okay. I'm not sure what this means. See? Maybe cyto something. NAD plus to complex 1 to ubiquinone to complex 3. And then complex 4 and then O2. If we involve complex 2, FAD from here goes to FAD, complex 2, and then through ubiquinone. It makes sense. Let me bring out the illustration for stage 4 of oxidative phosphorylation. Okay, so from complex 1 through ubiquinone to complex 3, complex 4, and what? Complex 4, and then to oxygen. Oxygen isn't shown here though, but it's somewhere here, definitely. FAD to complex 2, and then through ubiquinone. Complex 2 is right here. Okay, so FAD, complex 2, and then through ubiquinone, and then complex 3, and then complex 4, and then oxygen. Okay. And oxygen makes electron transport chain aerobic respiration. Like I said in the previous vid, stages 3 and 4, more particularly citric acid cycle and oxidative phosphorylation, both of them can function without oxygen. In other words, both are anaerobic. But because of oxygen here, it can make the ETC aerobic respiration. In theory, I think it has to allow for a faster processing of, well, it's basically faster to generate ATP if there's oxygen, I think. Because the policy, if there is no oxygen, I think if there is no oxygen, then the process should be slower because there's no ATP involved. I might be saying the wrong thing though. That's just in theory, but yeah. All you have to know is that oxygen makes ETC aerobic respiration. Note, 1 NADH yields 3 ATP molecules. 1 FADH yields 2 ATP molecules. Where do we see that evidently? I think it's seen here. Is it? Doesn't connect though. I'm not sure how it's connected. Anyways. Okay, let's move on to Calvin cycle and light reaction. Which comes first? Chronologically, I think it's this. Yeah, it's this, light reaction. That's the first stage of photosynthesis. And yes, might have guessed, I should have drawn an illustration here, but I didn't have time anymore. I could, but it's already the past. If I needed to study this again officially, then I would make this reviewer prettier. But yeah, this is the past. Where do we start? PS2, PS1. Essentially, light reaction is the first stage. It produces ATP, oxygen, and NADPH. ATP through phosphorylation. Photophosphorylation. Okay, my notes here are incomplete. Oxidized, loss of electrons, NADP+. Reduced, gain of electrons, NADPH. When photosystem is present, or in other words, chlorophyll pigments absorb light, pigments become oxidized, not reduced. ATP gives energy, while NADPH gives reducing power. Okay. That's it for this page. How about this? Okay. Cyclic electron flow does not produce NADPH, O2 produces ATP. Several photosynthetic bacteria only have PS1. For example, purple sulfur bacteria. Okay, but usually they have both PS1 and PS2. 3 steps in light reaction that contribute to the proton gradient. So we have water splitting, PQ. This is a mobile carrier protein. It transfers electrons to the cytochrome complex, which is this. 4 protons are transported through the thylakoid space. H ion is removed from the stroma as NADP+ reduces to NADPH. Both of these steps contribute to an H ion gradient in the thylakoid membrane for ATP synthesis. So it's similar in a way to electron transport chain and chemiosmosis. More specifically, chemiosmosis, because a gradient is produced and it allows for the triggering of ATP synthesis. Again, oxygen is highly electronegative. It's really good at attracting electrons. And I think that's why it's termed or coined as, let me just check. Let me just search that again. Final something. Final electron acceptor. Makes sense. It's really good at attracting electrons after all. Okay, that's it for this page. Fortunately, I won't be explaining that anymore. I don't remember. Calvin's cycle. So this is the second part. By the way, if you haven't figured yet, I think, this involves plants basically, how they produce energy. Okay. Citric acid cycle is catabolic, exergonic. Calvin cycle is anabolic, endergonic. Exergonic again is releasing energy. Endergonic is the opposite. So the Calvin cycle is, well, it has three phases: carbon fixation, reduction, and regeneration. So coming from CO2, uh, so it's short-lived intermediate, 3-phosphoglycerate. Each receive an additional phosphate group from the ATP. So ATP becomes ADP, and so we have 1,3 instead of just 3. 1,3 by bisphosphoglycerate. NADPH becomes NADP plus. I think this is an oxidation reaction, is it? A pair of electrons from NADPH reduces 1,3, whatever this is, and also loses phosphate group to be G3P. I think this is the same as this one. This. By the way, I'm talking about the 10-step process of glycolysis, G3P. I think this is the full name that's being referred. So regardless, this one is for plant cells. Glycolysis is the first stage of cellular respiration for eukaryotic cells. Technically, they're not so far from each other. They're not so different. So that's what I think about this. So phase 2 reduction, G3P. Yeah, I did show you. It's the same. Glyceraldehyde 3-phosphate. Or in other words, sugar. 3 carbons unlike. Okay, I did not continue there. That's sad. Okay, next step, or phase 3 regeneration. In the last steps, 5 molecules of G3P are rearranged to make 3 molecules of RUBP for the next cycle. So from here to here. And ATP is used for this. So ATP becomes ADP. The phosphate group is brought here, so that coming from here, it becomes this. And then Rubisco, I think this is an enzyme, I'm not sure. Okay. That's it. Can't explain better. Okay. For the net synthesis of one molecule of G3P, the cycle must happen 3 times. So consequently, there's a mix of 3 molecules of CO2. So one cycle is to 3 molecules of CO2. This is the standard. So 3 turns of the Calvin cycle. This is to fix enough net carbon to export 1 G3P. Each turn makes 2 G3Ps. So 3 turns makes 6. 2 times 3 is 6. And only 1 G3P leaves the cycle for other compounds. The remaining 5 stay in the cycle to regenerate other compounds within. So again, it's a cycle. Only 1 leaves out of the 6. What you have to also consider is that the cycle began with 15 carbons of carbohydrates in the form of 3 RUBP molecules. So this is the 5-carbon sugar. Now, there are 15 carbons of carbohydrates in the form of 6 G3P molecules, the 3-carbon sugar. So 1 G3P molecule exits the cycle to be used by the plant cells. The remaining 5, as stated here, gets recycled to regenerate the 3 molecules of RUBP. Okay, could have detailed better, but yeah. Okay, what's this? Photosynthesis. Okay, this is another take of the concept. So photosynthesis, we have 2 stages. One is light reaction, the second is Calvin cycle. So this is the equation of photosynthesis. ATP, energy or endergonic, it's endergonic, plus 6 carbon dioxide molecules plus 6 water molecules equals to glucose and 6 oxygen atoms. So this is just a byproduct. And yeah, reduced, oxidized. So from here to here, a reduction reaction happens. And from here to here, an oxidation reaction happens. Okay, so chloroplast uses light energy to make sugar by coordinating these two stages of photosynthesis. Okay, so for stage 1, what's the purpose? It is to produce NADH and ATP by phosphorylation. NADH as a source of electrons because this is a carrier of electrons. ATP is the energy currency of cells. Where is this located? Where does this stage happen? At the thylakoids of a chloroplast. So, where is that here? Yeah, I think it's this. Stage 2, requirements are NADH and ATP from the light reaction, the products basically of LR. And the purpose is to produce sugar. And where is this located? The Calvin cycle. It happens in the stroma. So it reduces the fixed carbon to carbohydrates by the addition of electrons. So this is carbon fixation. Reducing power provided by NADPH from collected electrons in stage 1. Okay. NADP plus picks up electrons here. ADP picks up an inorganic phosphate or PI. ATP is brought to the Calvin cycle as well as NADPH. As said here, as said here. So NADPH has electron cargo. That's the reason why it's not plus. Initially it was, but now it's not. On the way to the Calvin cycle. So it's a cycle, essentially. Backstage. Linear electron flow, cyclic. I think this, an example of this is CR or cellular respiration. I'm not sure. But based on the products of the linear electron flow, it says ATP, NADPH, and O2. Oh, okay. This might not be relevant. Both of these may only correspond to plants, but yeah. The only product of cyclic is ATP. Linear electron has that plus O2 and NADPH. Okay, I wrote these so that I might be able to distinguish them, but I guess I wouldn't now. Okay, what is this? Cell, lysosome, and nucleolus. All cells have genetic material, all have a cytoplasm, and all have membranes. That's standard. I think lysosome is, what do you call this? The garbage can in a cell. So anything that's dead and anything that's considered waste, the lysosome eats. Possibly maybe through endocytosis, I'm not sure. But yeah, essentially it collects the garbage and brings it out of the cell. The nucleolus, it's located within the nucleus. I believe this contains genetic material. So yes, that's it for this page. Okay, I think that's it. Is that it? I don't think so. Okay, yeah. We still have a little more. Functions of a cell membrane. So again, I did mention about this in the previous vid. It's here again, but I did not repeat. This is a different page. I guess I wrote it again when I was reviewing. Um, so the functions we have 6: transport, signal transduction, cell-to-cell recognition, intercellular joining, enzymatic activity, and attachment. So it's intended to regulate transport inside and outside of the cell. And again, it looks like, should I draw? No, let me just find the image again. Yeah, it looks like this, the cell membrane. So going out, going in. Active transport, there are transport proteins here. So if you have ATP and you have to go against the electrochemical gradient, you need ATP, and yeah, it's basically active transport. For example, you want glucose to enter the cell. So coming from outside, there has to be a carrier protein appropriate for that glucose molecule so that it could enter the cell. And it's fast because it's active transport. ATP is used. If it's passive, for example, you want to transport water, a water molecule, let's say it's going in, it only needs a specific transport protein, and I think that's called channel. This could be used for passive transport. So, but yeah, the process is slow. That's what makes it different. Okay. So that's for the first function. Okay, next. I don't like this. No joke, this is one of the lessons that I hated the most. Transport systems of plants. I never liked botany in general. Wasn't worth the hype, I guess. I preferred second quarter grade 12 biology, where we learned about the human body, diseases and stuff. But yeah, let me just... Should I make a separate video out of this? Maybe. I think so. I should stop this video now. But yeah, expect that I won't be discussing much on botany. But yeah, that's it. Thanks for watching.